My title is really about using large-scale simulation to converge reproducibly the distributions of conformations and dynamics of nucleic acids, and we're going to talk about two parts of that today, two projects, funded mostly by the NIH and NSF, and with lots of computer time, not only on Blue Waters, but also through Exceed and on the D.E. Shaw Anton machine at various times. And what we'd really like to understand is RNA dynamics and structure, and RNA is really a neat molecule because it's very sensitive to its surroundings and interactions with other solvent ligands that can change the structures. And multiple conformations can be populated that are biologically relevant, so it's a neat system to model. Examples are ribose switches that bind a ligand when it gets to a certain concentration, leads to a conformational change that shuts down the import of that ligand into the cell or whatnot. And it's a challenging system because of the high charge. You need accurate force fields. We need a good way to assess it. There's not a lot of great experimental results in some of these cases. And the dynamics are complex, and it's difficult sometimes to know if a structural change you see in simulation is real, representative experiments, or an artifact of the force field. And we're seeing some nice success. For example, there's various loop structures that are influenced by the binding of magnesium will convert from one structure to another. We can throw magnesium in a simulation and convert to the correct structure, but the reverse isn't possible, partly because the magnesium-free structure from NMR is not very well resolved. But we're going to show some bigger problems as we move forward. And what we've kind of seen over the last 25 years or so, gosh, I've been doing this for a long time now, is that if you run short simulations, you can stay very close to an experimental structure. As you tend to run longer, we tend to move away to some other structure that may or may not be representative. Ideally, that structure, if it's not representative, is going to have a higher energy, but we often see the structures that we find are much lower in energy than the native structure, and we get trapped there, and they're artifacts. And there's a lot of ways we can do these simulations. You could do a brute force simulation using D.E. Shaw's machine on dihydrofolate reductase, about 23,000 atoms. You get 16 microseconds of MD simulation a day. But an alternative is to use ensembles of independent simulations that may or may not be exchanging information to enhance sampling. And with the current AMBER GPU code, which is one of the fastest on a single GPU, with hydrogen mass repartitioning, we can get about 110 nanoseconds a day per GPU instance. So on the order of 150 GPUs will match the Anton performance if aggregating the results is equivalent to one long-time dynamics, and we'll show that it is in our hands. Okay, AMBER is a code that's been around for quite a while now. We just released AMBER 14. All the methods I'm talking about today, the analysis, the tools to run the simulations are available in the AMBER 14 release that just came out. And we had to add in support for multidimensional RMD simulations, which I'll get to in a bit, and all the analysis. And this is a little bit of a tricky thing because we've got lots of independent simulations that are exchanging information, but they're writing out a trajectory of the atomic coordinates that is swapping temperatures or Hamiltonian or other things. So you've got to go back and sort that. We originally tried to do that on a single node of Blue Waters using all 32 cores, and it crippled the I/O subsystem. So we had to parallelize our code. So now we're doing it fully distributed analysis in parallel with using a single core per node, and that allows us to process through this data really efficiently, really nicely. And then the AMBER GPU performance has increased considerably, and now also we can support peer-to-peer communication with multiple GPUs. So if you're fortunate enough to get a box that's got 4 GPUs in it, you can use all 4 of them for the same problem and get essentially a great speed up. Okay, so 2 short stories. One is, can we converge DNA duplex structure? And this is a long time project over the last 4 or 5 years. When I started out as an enthusiastic young assistant professor, I wrote lots of NIH grants about DNA work. And one of my favorite comments that came back, I never got funded for DNA work. One of my favorite comments that came back was, haven't we learned all we can possibly learn about MD simulations of DNA? And I said we hadn't, but we didn't. I moved to RNA because RNA is eminently fundable. Now we're coming back to DNA in some bit. Okay. So the longest simulations of DNA duplexes that have been published, to my knowledge, in the order of 1 to 4 microseconds of simulation. And on this time scale, there wasn't complete convergence of the backbone distributions or ions. we have been involved in this consortium called the ABC, to divide and conquer approach to look at tetranucleotide sequence repeats in DNA. We each did a subset of the simulations. This has been done over the last decade, more than a decade, 12 years. But one sequence had a bimodal twist distribution, so I thought it'd be fun to, at the central CG, so I thought it'd be fun to throw that on Anton and see what we could see. And the original ABC simulations were pretty short. We run a long simulation on Anton. We see some funny, with the RMSD, the initial structure is flat. We see some interesting bumps. Those bumps turn out to be terminal base pair opening, and they're occurring on about a microsecond time scale, which is consistent with experiment. And other properties like B factors comparing Anton to a standard Amber simulation were very consistent, or average structures where we look at 5 nanosecond average structures. The spaghetti movement of the atoms looks like we would expect, but the unexpected result we found in about, well, here's just a movie of that motion. The unexpected effect we found was when we created average structures over longer periods of time, like a 3 microsecond window, averaging the coordinates, and compared them from different times in the trajectory, the structures were essentially identical. They overlap with an RMSD that's really, really tiny, like 0.03 angstroms. So really, really close. And this was completely confusing to me because we know that DNA bends and breathes and has all these complicated twisting motions. And so the fact that it stopped moving on this microsecond timescale, I assumed was an Anton problem. So we went back, ran all these simulations on Amber GPU and CPU. Those took 2 years and only got to a few microseconds, not nearly as long as the Anton simulations, which we had out to 44 microseconds. So then we started to do ensembles of simulations. We had 100 independent simulations. We got access to the KIDS and Keeneland system early and friendly user time, so we could blast those out on the GPUs and show that the results were essentially equivalent between those and that this property is real. And we'll discuss it in a bit more detail in a minute. Now, if you look at a longer Anton simulation run, we see some bumps. These bumps are the base pair opening. Here, it's 2 base pairs opening and 1 on the other end, which leads to a larger RMSD, but it closes by the end of the simulation. And here's an ensemble simulation where we put 100 independent simulations together, throwing out the first 100 nanoseconds from each. And you can see it does base pair opening, but not as consistently as in the other simulations because these guys are longer. The Anton runs are longer. They can fully sample the event, whereas we may just catch an opening in each of these simulations which are on the order of 400 nanoseconds each. We developed some tools to compare simulations very easily. One is with principal components, where you combine the 2 trajectories and you calculate your matrix, your covariance matrix of essentially principal components, and then you project those eigenvalues, eigenvectors back on the individual trajectories separately. And those projections look like this, that the dotted versus the solid comparing the 2 Anton runs. And if you include the terminal base pairs, they don't overlap well because they're showing that we haven't converged the terminal base pair opening clearly. But if you get rid of the 4 base pairs on each end, you now see that the modes of motion are identical between the 2 simulations. They're both sampling the same modes. And these are the lowest frequency modes. 2 of them are shown there, which represent some bending and twisting. We've also developed this cool Kullback-Liebler analysis. Dan Rowe in my lab has done this, where you can now watch how these projections of the principal components converge as a function of time. And so you can see some of the modes actually don't even converge on an 8 microsecond time scale when you include all the bases because the terminal base pair opening events are occurring. But again, if you omit the 4 terminal base pairs on each end, by about 2 microseconds, you're down near 0, and you can argue whether you're by 1 to 5 microseconds, you've essentially averaged out all the motion in the system. And another way to see this is to look at running averages of a trajectory. So if you smooth over a particular time window, that gets rid of motions, and then you can cluster the trajectory to see the structures that are most diverse from each other. And this is shown at a 325 nanosecond window, 10 clusters representative of this trajectory. And you can see already by 325 nanoseconds, a lot of the DNA structure is pretty converged, except in the termini. By an 8 microsecond window, you don't really see any difference, except little fluctuations in the termini that represent differences in the structure. And, these simulations run with the latest Amber force field for DNA. Maybe this convergence property of the dynamics of DNA is an artifact of the force field, so we ran it also with the Charm 36 force field using the same ensemble calculations. This is kind of a measure of how well the structure is converging to the average structure. And Charm takes a little bit longer to converge, maybe more like 3 microseconds. That's because it does a lot more terminal base pair fraying, up to 3 or 4 bases in compared to the amber force field. But essentially the same results are seen. Now, does this make sense for DNA to essentially have a structure that is essentially rigid on a 1 microsecond time scale or beyond? And that leads to questions about conformational selection, induced fit, deformability, why are mismatches easily recognized in the DNA duplex? And if you look at the combined set of experiments, there's many experiments that measure a particular motion, like a certain distance that they can probe via experiment. And they show motions up to about the high nanosecond time scale. There's a few that show some in the microsecond. There's also nice work on dynamic Stokes shift, which shows a continuum of motion from femtoseconds up to about 40 nanoseconds. That includes the salt and the ions, and you can't break it up into any exponential fit. It's essentially a power law distribution of motion over that time scale. So we see lots of motion there. What about at longer time scales? Well, internal base pair opening is known by NMR to occur in a 5 millisecond time scale, 5 millisecond time scale. There's certain sequences that can do it about 1 millisecond or faster, but essentially there's no measures of motion in this particular range, and it's often been attributed to it's very difficult with experiment to measure in this range. But what if there is no motion in the DNA duplex in this range? And that's kind of our hypothesis now, that this gap is in fact real. An exception is some nice work by Al-Hashimi by NMR that shows with a lesion where you've got a damaged base pair, a signal suddenly pops up in this region where I said the dynamics are flat. And to me, this kind of represents like a siren, a beacon that says, hey, I'm damaged DNA. And this time frame of 1 microsecond to 1 millisecond, the encounter time of a drug or a protein with DNA is on the order of 1 microsecond or so. So essentially, the DNA is showing a rigid picture on the time scale of the encounter, so that the protein is really recognizing by conformational selection, not induced fit. If there's induced fit, it's going to involve base pair opening or kinking of the DNA, which can occur in a much longer time scale or via an activated process. So we've just submitted this to one of our more higher profile journals. We'll see if it makes it. It's taken me about 4 years to get this understood. Just quickly on RNA, I mentioned the problem. We typically run a simulation. It goes to something that's kind of not representative of experiment. A good example is UUCG tetraloops with whatever force field you want to run these days. But if we run a bunch of series of 100 independent simulations, you can see a bunch of them move far away from that starting structure, and they move to something that's not really representative, and they don't come back. This is the same on Anton, where they all run away by about a microsecond or so. And so we moved to very simple systems to begin with, the RNA tetranucleotide, because we can sample this fully. There's 2 structures that are proposed for this that are both similar to an A-form RNA, the major and the minor. The major is more populated than the minor, and these fit NMR data of Doug Turner. If we run a straight MD simulation, we actually sample different conformations, but we haven't converged, for example, the blue conformation. Also, there's more than 2 colors there. There's 4. We find 4 predominant conformations, this inverted structure and this intercalated structure, which are partly some of my nemesis at the moment we're trying to fix. Thanks to Blue Waters, and also before Blue Waters, access to Stampede, and, well, not Stampede, it was Keeneland back then, but Stampede's also got GPUs on it. We can use these ensemble calculations to really crank these things out. And what we do is we run independent simulations with Amber that are really fast in the GPUs, and we exchange information such as temperature, force field changes to make it easier to rotate around dihedrals or accelerated MD, or many other, and we can generalize to any number of dimensions in Amber at present. And I'll mention that the current players on our PRAC award also include Adrian Reuterberg, Carlos Simling, Darren York, who's upsetting me now because most of the jobs on the system that are under my name are his. They're doing QMMM calculations. And Dave Case we've added in recently. Now, if we do the replica exchange, we see a continuous distribution of blue, green, and the other conformations. We're fully sampling these conformational spaces. Very exciting. Just with temperature, using 24 replicas, it still takes about 2 to 3 microseconds per window to converge this conformational distribution, which is a lot for a little tetranucleotide. You can imagine if you're going to go to a tetraloop, it's going to be much longer than that per replica. And the way we look at the populations now are compared to A-form reference is 0. We can see the population a certain distance away in terms of RMSD space from the correct structures. Now, the correct structures are the major and the minor, which are not populated at the highest amount. These other conformations are populated higher. But we can do independent runs and show that they are exactly reproducible with different initial conditions in terms of the conformations and the ion placement and so forth. So it's exciting that we can converge these with Blue Waters. We can do it even faster with the multi-dimensional replica exchange where you use more GPUs, but hopefully for less time per window. And this just shows the overlap of the principal components for a multi-dimensional replica exchange run with a dihedral force modification and temperature, and the dotted and solid overlap pretty well. And we now, for all our papers, we put in the supporting information scripts how to actually analyze this data and generate these plots, principal component analyses, so others can figure out how to do it because it gets kind of complex, but the code's all available. And now with the multidimensional REMD, we're converging on the order of 100 to 200 nanoseconds per replica using 192 replicas, which is nice because the time to solution now goes down to, that can be done in 2 days, essentially, on Blue Waters. And we can look at Forcefield modifications. There's a DAC phosphate from David Case where they modify the phosphate that gets the major and the minor better. We're playing now with HMAS repartitioning, which is used in NAMD and GROMACS and other codes to allow us to go to larger time steps, and we're verifying that we're getting the same. We haven't fully verified we're getting the same kinetics, but we get the same conformational distributions twice as fast. And now we can compare lots of different RNA force fields and see the behavior. So for example, with CHARM gets the major and minor conformations better, except it flips the terminal base pair of SYN. It's got a misbalance in the glycosidic torsion. The Chen-Garcia, which does very well with UUCG tetraloops, completely messes up the tetranucleotide in terms of its conformational distribution. So there really is not the great RNA force field yet. We're trying to fix this, but we're not quite there. We now can converge the UUCG tetraloop. It's a more complicated system, bigger. You'll see in the beginning we're populating near the experimental structure, but we're gradually reducing the experimental structure in favor of some that are like 6 angstroms away and not representative, not real. And we think we have handles on what's going on. We know the amber force field tends to overstack a little bit, but it also has hydrogen bond strengths that are too strong as well. So certain these folded conformations put hydrogen bonding groups that just lock it up in various ways. And this kind of shows the convergence. The convergence now for the tetraloop is longer per window. We're now running not 192 replicas, but more like 360 or 500 replicas. And that each one of them has to run for on the order of up to a microsecond. So still pretty computationally demanding. We're very interested in sharing data. I'm going to talk with Alexi's group later today about their efforts in sharing this, but this is one of our efforts where we tried to develop an environment where you can automatically annotate your simulation data, put it up on a web page, and then make it available to the community. The complex version uses iRod's database to create these things, and you can configure your own XML to refer to particular files to display the results. This is great when you got lots of different force fields you want to test because you set up the analysis once, then run it across all of them and just look at the web page of the results, which I normally was doing by PowerPoint. It took me forever. So this is a much nicer way. And then CPP Traj is our analysis program. It works with many different trajectory formats, GROMACS, DCD, AMBER, and others, and can do a lot of complex analysis in a fairly straightforward way. If there's analyses you think you want and need that you want added, you can always ask us about that. Dan Rowe, who works in my lab, is just a phenomenal programmer, and it's been working really well. And with that, I will stop and ask 4 questions.